Welcome to our presentation on the molecular identification of the Crape Myrtle powdery mildew pathogen, a collaborative effort by researchers at Tennessee State University. Crape myrtle trees are known for their vibrant flowers, adding beauty to landscapes. However, they are susceptible to powdery mildew, a common disease affecting their health. Powdery mildew is a widespread disease in crape myrtle, with various pathogens identified across different regions, including the USA, China, Japan, and Australia. We explore whether the causal pathogen of crape myrtle powdery mildew is consistent across regions and how many pathogens are involved, given the challenges in identification. The internal transcribed spacer, its region, is crucial for identifying fungal pathogens in plants, with thousands of sequences available in GenBank for research and analysis. Our objectives include identifying the causal pathogen for powdery mildew in crape myrtle, developing species-specific primers, and detecting the pathogen using these primers. We employ a four-step procedure for its universal primer analysis, including DNA extraction, PCR amplification, sequencing, and similarity analysis. Genomic DNA is extracted using the DNAseq plant mini-kit from the mycelia and conidia of powdery mildew, a critical step in our analysis. Using its universal primers, we amplify a 704BP PCR product, a key component in identifying the powdery mildew pathogen. The sequence from the PCR product is analyzed to understand the genetic makeup of the pathogen, providing insights into its identification. Our sequence analysis compares different pathogens and their primer pairs, offering a detailed look at the genetic differences and similarities. Through blast search, we identify 100% identity with Uncineliella australiana in GenBank, confirming the pathogen's presence in crape myrtle. We design specific primers and conduct tests to verify their specificity, ensuring accurate detection of the pathogen. Our primer design process involves selecting sequences and locations to effectively target the pathogen, a crucial step in our research. The amplification pattern of DNA using 13 primer pairs helps us detect Uncineliella australiana, showcasing the effectiveness of our approach. Specific primers reveal the presence of Uncineliella australiana as the powdery mildew pathogen in crape myrtle, particularly in Tennessee. We compare crape myrtle powdery mildew with other pathogens affecting lilac and dogwood, highlighting the differences and similarities. Specific primers for E. pulchra in dogwood powdery mildew demonstrate the precision of our molecular identification techniques. Our research includes specific primers for P. guttata in dogwood powdery mildew, further expanding our understanding of fungal pathogens. The polymorphic band for Microsphaera syringae japonicae in lilac powdery mildew highlights the specificity of our primer design. In this study, we identified specific primers for Uncineliella australiana, the pathogen responsible for powdery mildew in crape myrtle. The image shows the polymorphic bands unique to U. australiana, highlighting its distinct presence among 4 DNA groups. Our research confirmed that Uncineliella australiana is the causal pathogen of powdery mildew in crape myrtle in Tennessee. We found that the polymorphic bands were amplified at 666 BP and 704 BP, allowing us to distinguish U. australiana from other pathogens in nursery crops. A question arises. Is Eryciphe lagerstromiae synonymous with Uncineliella australiana or Eryciphe australiana? This query opens the door for further investigation into the taxonomy and classification of these pathogens. We extend our gratitude to our supervisors, Dr. Margaret Emembaga and Dr. Suping Joe, and our dedicated lab team, including Dr. Emmanuel Nodu, Dr. Frank Marima, and Dr. Mario Carey. Special thanks to Dr. Nick Gall, Dr. Roger Sav, Dr. Sandra Reed, and Dr. Klopfenstein for their invaluable support. Thank you for your attention and participation in this presentation. Your engagement is greatly appreciated, and we look forward to further discussions and collaborations.